Yeah, Piyusha, can you hear us?
Yes, yes,
Amazing,
absolutely.
great. Right, so take us through, thank you.
Okay, so please here you have the floor.
Is it visible?
Yes.
Okay. Thank you so much. Good evening, everyone. I'm Dr. Piyusha Majumdar from India. And first of all, I'd like to thank WANIDA and ACE Partner for shortlisting my work and giving me chance to present my work in the area of implementation research. My topic is implementation challenges and evolving malaria control strategies from global strategies to local innovation in the tribal areas in India. Here, when we talk about India, so it's a country in the South Asian region, and we're the 7th largest country by area and 2nd largest in population after China. We all know that malaria is the oldest disease of civilization, and we have started our first national health program for malaria control in 1958. And it has been more than 60 years. Our country has made several progress in malaria reduction, but still we have not been able to eliminate it from the country. And in this presentation, I'm going to talk about what are the implementation challenges that are hindering the path of malaria elimination? What are the global strategies that were used and how we have used the innovative strategies in the tribal area to reduce the prevalence of malaria? And what more could be done to eliminate it and how contribute towards reaching our sustainable development goal? So let me give you the background of that. When we look at the latest World Malaria Report 2020, India contributes to only 2% of the cases. But when we look at the Southeast Asia region, we are contributing towards 88% of the cases. And the transmission in India, it's highly complex and, basically, because it is concentrated more on the tribal regions, the forest areas, and those areas which is majorly dominated by the marginalized population. And these, they are the areas like Madhya Pradesh, Chhattisgarh, Orissa, northeastern state, where the implementation challenges are the greatest. So in this slide, I'm showing the journey of malaria control program in India. So as you can see that, we have done a spectacular achievement from 1950 to mid-1960, because we have launched a global malaria eradication program of WHO. And the number of deaths has significantly reduced till 1960 when we have launched the malaria control program. But it was reintroduced and spread widely due to a large number of factors, like we have slow development of health infrastructure, surveillance was not adequate, logistical challenges, then there was vector resistance, and there was a low priority when it comes to the post-control phase. So in 1977, India has, they started a modified plan of operation in which we have created a 3-pronged strategy. And in this 3-pronged strategy, we have done a lot of government effort, we also done a lot of malaria research, and there was a need of public participation also. Now, in 1998, the program was renamed as National Anti-Malaria Program. In 2003, some more disease have been added, and like chikungunya, dengue, Japanese encephalitis. lymphatic filariasis, Kala-Azar, and the program was renamed as National Vector-Borne Disease Control Program. When it comes to 2015, we have taken, India has taken a pledge in eliminating the malaria in 2030, and we also launched our national framework for malaria elimination and made our strategic plan to eliminate the malaria by majorly focusing on the prioritized states and districts in a phase-wise manner. So this is basically, I've shown you the journey of malaria control program, which we, which is being evolved in India. Now, this slide is showing that how, the program and the strategies evolved from 1958 to 2017 by taking into account the epidemiological parameters. Let me come to the study objective. So my study objective basically is to assess the barrier or challenges in implementing malaria control strategy in tribal area. As I have mentioned that, the malaria control is, quite challenging and complex in tribal areas. So I'm going to discuss what are the barrier and challenges that are going in tribal areas. The innovative strategies of malaria elimination that we have used here in the tribal area. And beside this, I'm already suggesting here a theory of change to accelerate and achieve the path of malaria elimination in 2030, because we have taken a pledge to eliminate the malaria in 2030. The methodology which we have used here is an exploratory approach. I've reviewed the published and grey literature and the other research evidence to generate discussion. Reviewed and synthesized around 30 evidence and done a thematic analysis using ATLAS-TI software. So before talking about the programmatic implementation challenges here, I want to talk about the global strategy that that is being used in the program. So there was like mass drug administration, testing and treatment of malaria, and 2 rounds of indoor residual spraying and free distribution of long-lasting insecticide treated nets. And then there was the strategy of targeted drug administration, then targeted testing and treatment, and recommendation for malarial testing and treatment of organized or the people who have arrived from the malaria endemic areas. Then there's also a strategy on the recommendation for reactive drug administration, the case detection and treatment. So here, in this slide, I'm showing the consolidated framework for implementation research, and I've used, partly this framework in identifying the implementation challenges faced during the implementation of malaria control program in India. So as implementation research deals with identifying implementation problem, programmatic problem that hinders the intervention and delivery of the services. It also helps us in knowing which strategy work well in what circumstances. So if we know the implementation barrier in advance, it will help us in pre-plan the strategies so that our cost and time will be saved in the program, and will be able to reach the goal of the program. So this framework has 5 domains: inner setting, external setting, implementation process, characteristic of intervention, and characteristic of individual who are involved in the program. So now let me come to the implementation challenges. So in this slide, I'm showing the implementation challenges with regards to 2 domains: implementation process and intervention characteristic. Now this is further categorized into 4 major categories: the operational challenges, the operation challenges, the logistical challenges, the challenges which is related with the human resource in health, and the health system related challenges. So in some places, the major operation challenges was, use of indoor IRS. So there was like poor quality of IRS have used, and it leads to insecticide resistance. And as a result of this, we have not been able to, the cases have not been reduced as we intend to. Then another challenge was lack of community involvement. So when the program was launched, the community the participation of community was very less when it comes to MDA, Master of Administration, or even the advocacy activities. Another logistical challenge that we faced is reaching the unreached or the marginalized tribal population who are secluded, and moreover they do not have the adequate necessary infrastructure, and as a result of this, the service providers, there was unavailability of the service providers at the various health care facilities, and it leads to the human resource of health challenge due to infrastructure and lack of manpower. There is a delay in conducting microscopic examination of smear collected at village level. Then there is a delay in the diagnosis of malaria because there was longer time needed to transport slides to the laboratory and get the results. So there was inadequate surveillance also. So, all these factors are the implementation challenges. So in this slide, we have also tried to find it out, the implementation challenges in the inner setting. And we have again, it is again categorized into, individual, interpersonal, and organizational barriers. So when we talk about the individual barrier, the ITN use was affected by the cultural practices. People didn't know, they have lack of knowledge and awareness on the disease and its prevention when it comes to the tribal population due to their lack of education and lack of awareness. They didn't know how to use ITN, or due to their cultural practices, they were using it for some different work like fishing, and they are using it as a fishing net. Then there are interpersonal challenges. The healthcare, the frontline workers, they are doing a co-delivery of the campaigns. So as a result, the priority was different. The priority was some other program when it compared to the malaria program. And talking about the organizational barrier, there were like vacant posts of the officials, because there were a lot of insurgency in those tribal areas, and health service providers are not willing to come to the health facility. So as a result of this, there were many posts which is vacant in the program, and people were not willing to work. So this slide is showing the implementation barrier with regards to external setting. And we have further categorized it into 4 categories. So one is the social factor, another one is the second one is the community level factor barrier, then political factor barrier, technological barrier, or environmental factor. So coming to the social barrier, so the people have the belief in the spiritual cause of the physical symptoms. So there were inadequate surveillance and treatment, and rather than belief in the doctors, they were believing in the cultural healers. They thought that the disease is caused due to some other reason rather than malaria. So the community had very poor knowledge about malaria, and moreover their poor adherence towards the medication also. Another challenge that the community faced was there were unlicensed medical practitioners who were treating the patients. When it comes to the political factor, there was lack of political will and ownership, and they have competing public health priority. Talking about the technological barrier, there was lack of ecological and GIS-based database on the case and vector that impact this surveillance and the tracking of the cases. When it comes to the environmental factors, the tribal areas have vast evergreen forest and reserve that is covering 70% of the land area. and they have the climatic conditions that
Yes, sir.
favor
Can you
mosquito
make it faster,
breeding.
please? We are running after time.
Hello?
Can you make it faster? Yes.
Okay, sure. I'll just make it quickly. Okay, so when we talk about the barrier of the tribal population, so the barriers are poor housing, outdoor sleeping habits, and the tribal knowledge is poor. I've already discussed about that. The tribals practice doom cultivation. ITN were used for different purposes like fishing. There was non-adherence to the treatment regimen and unlicensed medical practitioner treating rural tribal population. Now these were the challenges, and our government has they worked on these challenges, and they have developed some innovative strategies to eliminate malaria by 2030. And I'm talking about the 3 innovative strategies that were used in Orissa and Madhya Pradesh. So the first one was Daman, and the second was CCMP and Malaria Elimination Demonstration Project. So most of these innovative strategies, they deal with eliminating the malaria in hard to reach population, and they wanted to eliminate the malaria by improving the access to diagnosis and treatment in hard to reach area. So in Daman project, the 3 major strategies that was used was routine, increase the surveillance and the case diagnosis and treatment. So it was used during a camp-based approach, and they also felt the importance of the community participation. So a lot of community engagement was done through ASHA worker, and they were prioritizing mainly the pregnant lactating mother and the under 5 children to reduce the mortality and morbidity among the vulnerable group. In CCMP, the major strategy that were used is they have strengthened the supply chain management because there were like frequent stockouts. So that's why they thought that it should be strengthened. Then they've also appointed block level manager to improve monitoring and supervision. As a result, there was a delay in blood smear examination. So they've established additional microscopy center at the primary health center level, so that the results will be faster, and they'll know that people are suffering from malaria or any other diseases. They for each and every case tracking, they have also done data digitalization and giving a unique identification number. The local volunteers have been they have been chosen and engaged in that. They've also done the intensified training for the and refresher course and capacity building for the frontline workers. The other innovative project is treat and track in Madhya Pradesh. So here also there are a lot of innovative strategies have been used. First is, a lot of work has been done on the communication material, IC or behavior change communication. They have developed the original material for their language only, so that community can use it.
Sorry, we had to stop the presentation because it is going. We're running after time. So thank you very much. We go to the next presenter. We have an oral presentation. Is he around? The next presenter. The next presenter. Aminu Yusuf. Yes. Yeah, so have you shared your slide?
I think it's already.
Okay, give me a second. I introduce Aminu Yusuf. So, let me. Aminu Yusuf. So, Dr. Aminu Yusuf from the St. B. Ahmed Bello University is going to talk about vaccine encoding trypanosomiasis brucei major surface proteins. So, you have the floor.
Thank you.
Okay, you can go with this one. Yes. Okay, thank you.
Okay. Good evening, everyone. I can see we are all tired, so I will be a little bit faster. My name is Dr. Aminu Yusuf Bashir. I am from ACE for Neglected Tropical Diseases and Forensic Biotechnology, Ahmadu Bello University, Zaria. And I am also a researcher with the Nigerian Institute for Trypanosomiasis Research. The topic of my discussion today is DNA vaccine encoding Trypanosoma brucei major surface protease B induces IgG response and controversial protection in immunized VABC mice. African trypanosomiasis, as we all know, is a debilitating disease that affects both humans and animals, and the distribution is in about 36 African countries, even though now the cases have dropped significantly from 10,000 cases in the last 10 years to last year, we have only 565 new cases. The disease is caused by African trypanosomes. For HAT, which is also called sleeping sickness, the disease is caused by Trypanosoma brucei gambiense in West and Central Africa, and by Trypanosoma brucei rhodesiense in East and Central Africa. In Nigeria, the last case was reported in 2012, but we have some suspected new cases in some part of the country. The vector of the disease is called Tsetse fly, and the parasite has a digenic life cycle, one part in Tsetse fly and one part in the vertebrate host. The antigen, the whole this work, the idea is to develop a DNA vaccine, and the target antigen for this work is called major surface protease. This enzyme is very important in the fact that trypanosomes are extracellular, and during their life cycle in the host, they encounter a lot of challenges, immune response by the host, and they successfully evade that immune response through what we call antigenic variation by switching one BSG to another. Now, for the new BSG to be expressed, the old one has to be removed, and it is this enzyme, major surface protease, that catalyzes the reaction that removes the old BSG so that the new one can be expressed. That is why I targeted it as a target to develop a DNA vaccine so that it can block this switching from one BSG to another. Chemotherapy is besieged with so many problems. All the drugs are very old. The trypanosome parasite have developed resistance against most of them. But the good news is that last year we have a new drug for the first time in over 80 years, fexinidazole, and furumidine and acrozibolosis are new drugs that are currently undergoing clinical trial. There is no vaccine for African trypanosomiasis. I will quickly go to the major objective of this work was to produce a DNA vaccine using full-length Trypanosoma brucei major surface protease B and evaluate its potential as a vaccine candidate in VABC mice. So this is the experimental design. First of all, this TB brucei strain is a local strain that is native to northern Nigeria. So we will extract dynamic DNA from the parasite, and then we isolate and provide the Trypanosoma B, major surface protease B gene from the genomic DNA as a template. then this gene was subcloned in PCR for topo vector, and then the recombinant vector was characterized by sequencing and restriction digestion, and then it was then cloned again in a modified PBAX plasmid. Then this was purified and then used as the vaccine candidate. And it was before immunization with mice, the vaccine was expressed in Hep G2 mammalian cells. So the gene was amplified by PCR using PFU high fidelity polymerase, but because this polymerase produce blunt end PCR products, so we had to add an A overhang to enable us subclone it in the PCR for topo vector using TA cloning. So this is the result of the PCR, and the gene has a size of 162 base pairs. So we now go and then clone that gene after purification and in PCR for topo, and this is the cloning technique. And then we did colony PCR, and then we selected the positive colonies, and then after that we purified the plasmid that contained the gene that was the MSPB gene. Then we did mini culture and plasmid purification, plasmid digestion, and glycerol stock. And then at the end, and then we now digested the plasmid in preparation for subsequent cloning. So we now go ahead and did cloning into this PBAX vector. This PBAX is a special plasmid which was donated by Professor Kem. And Professor Kem, I don't think he is here, he was here earlier on. So he donated this plasmid from University of Bremen, Germany. So this PBAX was restriction digested with SPEAR1 restriction enzyme and ECO-R5 restriction enzyme. This is the digestion, and then the gene was then inserted, and at the end of the cloning, we have the PBAX TB MSVB construct as our DNA vaccine. Then when we now sequence the full gene, we now do the blast N, and then we confirm that the gene is exactly as in the reference trypanosoma, which is Trypanosoma Edinburgh 927. So with these results, now we have our DNA vaccine. We now went ahead and did immunization trial in BAB-C mice, 30 mice, through intradermal injection. We have 4 groups. Groups 1, the animals were immunized with the DNA vaccine, and then they were challenged with the parasite. Group B, they were given empty PBAX plasmid, and they were challenged with the parasite. And then group C, group 3 were given PBS, and then were challenged with the parasite. And then group 4 were given only PBS. So this is the immunization schedule. So in day 0, we collected blood from the animals, all the animals in the group. This blood was used for ELISA, for IgG and interferon gamma assay, and then the other hematological parameters. And then on the same day, the animals were primed with the vaccine at 100-gram dose. 3 weeks after, we give them a first boost. 3 weeks after the first boost, the animals were given a second boost. And then on day 56, we collected blood again for ELISA and for hematological parameters analysis. And after that, on the same day, the animals were challenged with the 2,000 parasites. So we assay for IgG in all the group of the animals, and we can see through by ELISA, and we can see that the animals here in blue, which are the immunized animals, had more IgG response compared to other animals in the other groups. For interferon gamma, there was no any significant difference in all the groups, but you can see the immunized animals had a little more interferon gamma induction compared to other groups of animals. So we now check parastemia, and all the animals developed infection after parasites inoculation, and with a prepatent period of about 4 days, and eventually all the animals died, none survived. However, if you look carefully, you can see the animals immunized with the DNA vaccine had a lower parastemia compared to the other animals that were not immunized. And this lower parastemia is about 75% in the first week of infection and 51% by the 5th week of infection. So this is important finding, which has now encouraged us to develop a strategy that can confer permanent protection so that the animals will survive. But you can see there is about 20 days gap. So the immunized animals survive 22 more days than the animals that were not immunized. So this is an encouraging result. This is a Kaplan-Meier survival plot to show the percentage survival of the animals. We can see the animals that were not infected, that is group O, they all survived 100%, while the animals that were not immunized but infected, all of them 100% died by day 36, while the animals that were immunized, 38% survived up to day 58. So in summary, I will say the full gene of Trypanosoma brucei major surface protease B, which is a native of northern Nigeria, was sequenced, and the nucleotide sequence was found to be exactly as the reference Trypanosoma brucei Edinburgh in the Tribe3 data bank. And the gene was subsequently subcloned in PCR for TOFO, and then further cloned in PBAX plasmid to produce the PBAX TB-MSV construct as the DNA vaccine. When this DNA vaccine was immunized in animals, it induced significant IgG response, but non-significant interferon gamma response. Immunization with this construct was able to lower parastemia, as I mentioned earlier, by 75% and 51% on the first week and the 7th week of infection. And these translate to 61% improved survival period. These vaccines appear to be safe for now because none of the mouse died during the about 8 weeks of vaccination. So in conclusion, we say we were able to demonstrate how to produce DNA vaccine using the TB-MSV gene as an antigen, and then the results suggest that this gene can be used as an antigen in DNA vaccine development against African trypanosomiasis, since it could induce humoral response through induction of IgG and confer partial protection by increasing longevity and low parastemia. During the course of this work, we had a lot of limitations. For example, we are only able to do one immunization trial, and it's laboratory-based, not field trial. Only one route of administration was used, which was intradermal. I didn't use adjuvant during administration of the vaccine. and only one species of laboratory animal BABC mice was used, and only one dosage was used, was 100 microgram. So I recommend using a heterologous prime boost approach, whereby you can prime with the DNA vaccine and then boost with the protein, recombinant protein, and vice versa, and then to address all these limitations. And we are lucky we have secured a grant to address some of these limitations, but we are still looking for more grants so that we can improve the immunogenicity of the vaccine. So thank you very much. So sorry, before I go in acknowledgement, I want to acknowledge African Center for Neglected Welfare for sponsoring this work, Ahmadu Bello University Zaria, and my institute, Nigeria Institute for Trypanosomiasis Research. And then this work was done in Japan at the Institute of Tropical Medicine and the Institute of Global Health and Medicine, University of Nagasaki. So I appreciate all their support, including all the staff in Ahmadu Bello University Zaria and the Center for Biotechnology Research and Training, also in ABU Zaria. Thank you.
Okay, so thank you very much, Doctor. Thank you very much, Doctor. So could take one or two questions maximum. Do you have question around the audience? Any question? Yes.
Thank you for the talk.
Thank you.
My question is, do you think that the approach to boost during the 8-week of your study can give you a better protection?
Well, the boost was not for 8 weeks. The entire vaccination period, that is the priming, the first boost, and the second boost, that was 8 weeks. So the first priming was on day 0, and then 3 weeks after, we now give the second dose, and then 3 weeks after, we give the third dose. The idea is that the priming will sensitize the animal to produce the primary antibody response. But as you know, the primary antibody response is usually low and it doesn't last long. And that is why you give the second boost and the third boost, so that you are able to sensitize the animal for the secondary response, which is live much longer and is more bigger. So that was the reason for that approach. Some people vaccinate 2 weeks, but we decided to do 3 weeks. But because if you look at the literature, the primary response usually lasts up to 3 weeks. So that is why we choose 3 weeks.
Thank you. So, is there any questions? No more questions? So, okay, applause for...
Thank you.
Thank you very much. Asha. So we will call our next presenter, who is Asha Ashim Agolinu, PhD student, CA Tech MTVRS. So Asha is going to talk about the design in silico molecular model to study the SIF amide gene function in Anopheles. So since we're running after time, we would like every presenter to take maximum 10 minutes, no more. Thank you.
Thank you. My name is Maouyon Agolinu. I'm PhD student at the Nazi Bonin University, ICEM TV, and I'm honored for the invitation of WANIDA to participate to this event and in the field of strategy for vector-borne disease control and elimination. My presentation will focus on in silico design of molecular model to study the SAFMI gene function in Anopheles gambiae olfactory system in a perspective of genetic control of the vector. Mosquitoes are described among the most dangerous animals for the human. More than 3,000 species are identified, but only a few are responsible for the most dangerous disease transmitted to humans, with Anopheles caused around 700,000 deaths in Africa. In face to that, the control measures such as RSS, the LINS, the SIT, the destruction of the vector have contributed to the diminution of the burden of the disease, but have shown progressively the limits with the appearance and the persistence of the resistance to the diverse class of insecticide used in the control. So, the exploration of new approach is become important to overcome the disease in our country. Fortunately, the vector have a weakness. It uses its sense of olfaction to locate human hosts on which take its blood meal. So the odor are capped by the antenna, the massia part, and the labella, and processed in the brain of the insect. And so many genes are involved in this process. And the work of Antonia and the work of Martelli have shown the involvement of SAFMI neuropeptide in this process in Drosophila melanogaster, favoring the consumption of the food. So what is about the Anopheles gambiae? And in our study, we wanted to understand the function of this gene in Anopheles gambiae mosquito in case to disrupt the process of the host seeking of the insect. So to do this, we set a general objective, which is to develop in silico strategy to study the function of the SAFMI gene Anopheles gambiae, and specifically it was questioned to confirm the autolog of SAFMI gene in Anopheles gambiae and design in silico a knockout model of the autolog identified in Anopheles gambiae. Our study was in silico experimental study conducted in Institut de Recherche Ancien de la Santé, Burkina Faso. And the data we use come from database we consult such as NCBI, vector-based, fly-based database, which contain the annotated genome of arthropod, include Anopheles gambiae, which is our study organism, and Drosophila melanogaster, which is the reference organism we use in this study to analyze our results. So, for the identification of the autolog in Anopheles gambiae, we use ensemble metazoa database, where in the research box we fill the name of the gene. And to confirm the autolog we identify, we blast the nucleotide sequence and protein sequence of Drosophila melanogaster on the whole genome of the mosquito Anopheles gambiae. So we look for the protein concept domain by using the online tools, NCBI concept domain, and we check the expression of the gene pattern on Drosophila melanogaster by using Fly Atlas 2 and Moz Atlas for Anopheles gambiae. Also, for the knockout model of the autolog we identify, we choose a guide RNA by using the chop online tools for the CRISPR-Cas9 technology on the organism Anopheles gambiae. And we import this data on BenchLean tool. And we went to European nucleotide archive to select a plasmid which it use in insects and having several cloning sites, selection marker and a fluorescent marker. And we import this data also in bench-link tool for the construction of the specific plasmid in our study. So, in terms of results, the use of ensemble metazoa database allow us to identify one annotated autolog in Anopheles gambiae genome, which is AGAP 007056. In the genome of the mosquito, the gene is located on the chromosome 2 and have 2 exons and a code for 1 transcript. And the alignment of amino acid and nucleotide acid of the gene have shown in similarity more than 64% and the identity of more than 85%, and the gene appear to be well conserved in several Anopheles mosquitoes such as Anopheles gambiae, Coolisi, and Arabiensis. However, no conserved domain were identified on the gene. This should be because of the small size of the gene or a lack of annotation, or maybe the gene do not have protein conserved domain. Talking about the expression pattern of the gene appear to be expressed in the head of the both Drosophila melanogaster and Anopheles gambiae in male and the female. And those results allow us to say that the self-mint autolog in Anopheles gambiae is AGAP 007056. The use for the knockout model, the use of a SHOP tool allow us to select one guide RNA which target the exon 2 of the gene. And the genomic region which the guide RNA target appear to be well conserved among several Anopheles mosquitoes such as Anopheles gambiae, Anopheles merus, Anopheles quadriannulatus, Anopheles melas. And knowing that the endonucleasic activity of the Cas9 is to induce double-strand break of the DNA 3 nucleotide before the PAM site, then our gene will be cut at this position, and this allows us to construct right and leaf homology arm and entering in the 2L chromosome and have a size of 1,500 base pair and the primers to amplify them. And at the end, the plasmid we construct for our study have a basis feature such as the right homology arm, the leaf homology arm, 2 ATTP sites, the sequence of the fluorescent protein and its specific promoter, and we have also one origin of replication and marker of selection. And a plasmid with a same feature is used by Amond and the collaborator in a study where they study a CRISPR-Cas9 gene drive system targeting female reproduction in the malaria mosquito vector Anopheles gambiae. And the genomic edition can be confirmed by the primers in the table below. So, in conclusion, we can say that the autolog of self-made in Anopheles gambiae is AGAP 007056. The knockout model we designed have shown good efficiency as proved by the e-value less than 0.05 obtained in our construction. However, many ways remain to be explored to elucidate the molecular process of self-made mediate hot-seeking behavior in mosquito. The next step of this study is to continue in vivo investigation to confirm the effectiveness of the models we have developed in silico, and the probable application of this work is to develop genetic control tools such as gene drive to disrupt the host-seeking behavior of the mosquito, and to develop a bed for mosquito trap or improve the insect repellents. I can't finish this presentation without thanking all the collaborators which permit us to achieve this work. Thank you for your kind attention.
Thank you very much, Doctor. Maybe we can just take one question from the audience. Is there any question? Okay, so I think the discussion. Thank you very much. A round of applause for Doctor. Thank you.
Thank you.
We would like to call our next speaker, Rejoice. You have your presentation? You have shared your presentation?
Yes.
Okay. So Dr. Rejoice is going to talk about amoxicillin-resistant salmonella. Dr. Rejoice? One second. You have shared your presentation?
Yes, I've given it to Madam. Okay. I can start there.
Yes, you have the floor.
Okay.
Go ahead. Yeah.
Good evening once again. I'm talking about amoxicillin-resistant Salmonella enterica isolates from ready-to-eat meat sample in Ghana. When we say Salmonella, what is it at all? Salmonella is a bacteria, it's a pathogenic bacteria that causes typhoid infection. It has two main species, that is Salmonella enterica and Salmonella bungarii. It's the Salmonella enterica that causes the typhoid infection. This bacteria is always found in the excreta of either human or animal. So anytime this bacteria is seen in food, it means that the food have contact with either the excreta of man or animal. And globally, this is causing a lot of mortality and mobility. Globally, this disease, considering foodborne diseases, Salmonella is causing a lot of death, a lot of hospitalization, a lot of failure in treatment. And in Ghana, when we are talking about increased rates of the cases, Salmonella cases in the world, and Ghana is not exempted, every year there is a trend analysis that confirms that on yearly basis there is an increased rate of cases at various hospitals. Okay. The multi-drug resistance Salmonella enterica have been found in ready-to-eat meat. And this is a very serious problem because this ready-to-eat meat, they are not cooked again before consumption. It means that when you get the meat, you just eat it without consumption, without application of heat to it. And it's a major concern when this bacteria is found in food. and documenting antibiotic-resistant gene in ready-to-eat meat. From ready-to-eat meat, it's very, very important, but few researchers that focus in this area in Africa, hence this study. Materials and method. How is this study done? Samples were, the study was conducted in Accra metropolis, and the various sample collected, the Accra metropolis have been divided into 5 major markets. That is Malata, Agbogloshi, Kaneshi, Mamobi, and Nima. These are the places the samples were collected. And beef sample, mutton, chicken, spices, swab from the knife and also from the palm of the sellers were collected from several vendors. And in total, 360 vendors were contacted, samples were collected from them. These samples were sent to the lab, aseptically cultured, and we follow method described by Wallenson Ita 2019 in Bacterial Analytical Manual to isolate the Salmonella bacteria. That is, we follow pre-enrichment, enrichment, and then selective isolation. That is, when we talk about the pre-enrichment, the samples were kept in buffered peptone water, incubated at 37 degrees Celsius for 24 hours, and then 1 ml was pipetted to the Rappaport vacilladis, that is the Arvin broth, and also incubated at 41.5 degrees Celsius. From there, and a loop was what? Placed in it and script. Okay, and this so this bacteria was cultured in XLD, and then that is how it looked like on XLD agar. Looked blackish with the medium being pinkish. And then these isolates were also exposed to several antibiotic decks, as we can see it on the PowerPoint. We followed this diffusion method in doing this text. So the discs were placed on it, and then the zone of inhibition measured. The zone of inhibition measured. As we can see, you can see that some of them were, the bacteria was not able to, the antibiotic was not able to clear the bacteria around. Some were able to do something. Results. Looking at our result, you can see that from Agbogloshi market, out of the 72 samples collected from that area, 5 were positive. And beef sample, there is 1 positive. Chicken, mutton, from Agbogloshi market, it's only knife that we did not get any positive. When Kaneshi, Malata, or these 2 places too, we had 1 positive each. And then when we come to Mamobi and the Nima, we have 0 positive. So in all, only 7 samples that were positive of Salmonella enterica. So this 7 positive, when we ran it to percentage, 29% were coming from ready-to-eat beef, and then the spices used in preparing the sample, or when they are selling the sample, the spices that they add to it for us. But on knife, there is no bacteria or no positive sample have been identified on the knife. So when we look at the sensitivity test, when we look at the sensitivity test, you realize that amoxicillin was the only antibiotic that's okay. Amoxicillin was only antibiotic that wasn't able to clear any bacteria at all. Any bacteria at all. But looking when we talk about the susceptibility, those who are able to do something, you see ceftriaxone, ciprofloxacin, and so on. But in this case, what we are talking about is amoxicillin resistant. So this means that when this particular bacteria, when anyone is affected with this particular bacteria, when amoxicillin is taken, the person will not recover unless ceftriaxone and any other one. So this tells us that ready to eat meat in this environment, Accra Metropolis, particularly the Agbogbloshie market, is really contaminated with Salmonella enterica. So the vendors have to be educated on the way of handling the meat so that there won't be any contamination or recontamination. So when you eat meat around that place, there is a risk of consuming amoxicillin resistant Salmonella isolate. So our meat sample should be properly handled. Thanks for your attention.
Thank you, Rejoice, for your bright presentation and raising a very important question about animal and human health. So, do we take one question? If you, yes.
Thank you, Dorothy, for that interesting presentation. What could have been the source of this contamination? I believe this meat coming from probably the same abattoir. And from your research, you realize that only one market that you found positive sample for Salmonella. What could have been the source of the infection or contamination?
Please come again. Please come again. Yeah, the source of contamination could be from the abattoir. That would be the first source, the slaughterhouse, where they maybe as they are processing the meat or they are dressing the carcass at the abattoir, the GIT might have splashed on them. That would be the first source of contamination. And then the second source is when they themselves are processing, because much heat is applied to it. When I went to them, I realized that they use a lot of heat because the meat is very dry. So it can be recontamination after they have killed it with heat. If they themselves didn't wash their hand well, or sometimes the environment. Thank you.
Yes, so thank you very much. A round of applause for our presenter. Yeah, before I call our last speaker, I just would like to highlight this relationship between drug resistance between human health and animal health and even the environment, what we are calling now the One Health. Generally, we use antibiotic for feeding animals, and we don't pay attention that these animals have to take a time before they clear the antibiotic. And then animals are sold in the market, and we go to eat the this myth. So don't be surprised when you have antibiotic resistance for yourself and you didn't take any antibiotic. But also you know that most of us, me first, we are not adherent to treatment. I mean, when you take a drug and you feel that the symptoms are gone, you think you are healthy now and you stop taking your medicines. And this is one of the cause of developing drug resistance. So thank you very much for raising these questions. We are going to call our last speaker, Dr. Peter Yenugi Mahu, PhD. Dr. Peter is going to talk about the seroprevalence of dengue virus and co-infection with malaria and typhoid in pediatric patients in some hospital in Cameroon. Thank you. You have the floor.
Thank you.
Yes.
Okay. All right, I'm Mabo Peter Yunenui from the African Center of Excellence for Neglected Tropical Diseases and Forensic Biotechnology, Ahmadu Bello University, Zaria, Nigeria. One of the infectious diseases in this area of global change is dengue. We'll be looking at the seroprevalence of dengue virus and co-infection with malaria and typhoid in pediatric patients in some hospitals in Cameroon. Dengue, otherwise termed the breakbone fever, is that infectious type of a disease that is caused by the dengue virus. There are various serotypes, serotype 1, 2, 3, and 4, and quite often when you are infected with the virus for the first time, you can acquire a long-lasting immunity. But when you happen to be infected with another serotype, it becomes extremely severe. You can develop dengue hemorrhagic fever or the dengue shock syndrome. The breakbone fever, or dengue, is transmitted primarily by the Aedes aegypti species of mosquito, and secondarily by Aedes albopictus, though other species like Polynesiensis and Falcifer equally contribute in the transmission of the disease. Dengue is globally a public health problem and constitutes the leading cause of morbidity and mortality throughout tropical and subtropical regions of the world, with about 3.9 people being at risk. The diagnosis of dengue infection is rarely undertaken on routine basis in most of our hospitals, as the disease is generally considered neglected. And of course, many febrile illnesses due to dengue may be misdiagnosed for malaria or typhoid, and why not viruses like chikungunya, Zika, or the West Nile virus. In Cameroon, like in most sub-Saharan countries, dengue may concurrently occur, or they may coexist with other infectious diseases whose symptoms often overlap, thus rendering differential diagnosis a challenging one. The objective of this work were to determine the seroprevalence of dengue virus in pediatric patients, and also to investigate the existence of co-infection of typhoid, malaria, and the dengue in the pediatric patients. To achieve this objective, we carried out a cross-sectional study within for 6 months, that is from November 2021 to April 2022, in 2 public and private hospitals. We sampled first in the northern part of the region. If we look at this area, this is the study area. Adamawa region, Cameroon is made of 10 regions, and we decided to take this little cluster, this Adamawa region, and this is the central region where Yaoundé is the capital. And as I've just mentioned, we took 2 hospitals here, 1 private, 1 public, and equally 2 here. That is l'Hôpital Central de Yaoundé and l'Hôpital de la Caisse. In this study, a total of 477 consulted patients were recruited based on high fever that is less than 7 days. With this, with respect to the first objective, the blood collected from the consulted patients were being centrifuged and used to run a rapid test that determines 2 antibodies, IgM and IgG, as well as the non-structural protein that is unique for flaviviruses. So that is what we use to determine the seroprevalence of dengue virus. And second, next, with respect to the second objective, which was to investigate the existence of co-infection of dengue, typhoid, and malaria in the febrile patients, we did the same thing by obtaining the serum and testing for the dengue. On the other hand, whole blood was used to rapidly test for the presence of the histidine-rich protein and lactate dehydrogenase that is unique for both Plasmodium falciparum and the other species like Ovale, Vivax, and malaria. The serum equally was used to run wider agglutination tests, and the results showed that in Ngaoundéré we had 27.25 prevalence rates as opposed to 18.24 in Yaoundé, and an overall prevalence rates with respect to IgG, this rate is with respect to IgM or IgG or NS1. So any of the samples that were positive for any of these were grouped, and the prevalence rates calculated as such. What equally drew my attention, or what we can see from this result is equally the acute infection, that is always being predicted by the presence of IgG or the NS1 protein. Because for IgM, either the individual is exposed or it can equally be secondary infection. We can see a prevalence in Yaoundé of about 16 point something percent, and this result is comparable with that of Chuandom, who worked on the seroprevalence of dengue virus in children, but obtained a similar rate of 14.4%. So it means that the infection is actually active in Yaoundé. With respect to the co-infection, we obtained double co-infection as well as triple co-infection. For the triple co-infection, those infected with dengue, malaria, and typhoid was about 6.08. In conclusion, dengue virus seroprevalence appears to be high in pediatric patients in our study area of Yaoundé and Gaoundéré. Thus, the need to include differential screening for dengue infection in the management of fever. Thank you.
So thank you very much. Maybe one or two questions from the audience. Yes? Questions? Yes? Let me see if there is a question online. No question online? Okay. So thank you very much. A round of applause for our speaker.
Thank you, thank you.
Thank you very much. Thank you everyone for, I know everybody is tired now. But thank you very much for the exchange we have had along the day. And there are many interesting questions that have been raised and can be discussed after this session. So thank you very much. Thank you, everybody. Do you have a word? Sorry.
Coffee break on your way.
Yeah. So you can. We have the coffee break that's on our way. Thank you, everybody. Thank you. Let's say to tomorrow. Sorry. Yeah, tomorrow we have still the next session here in this room. Thank you very much.